nutrition and growth, population, the population formula, the factors that can influence growth. 50% come from control and all elements of control, except not antibiotics. There won't be any antibiotic stuff. I'm gonna take them all out. And then the 30% will be DNA and RNA biology. Because it's gonna be multiple choice, I have to figure out a way to be able to do this. So there'll probably be images and you'll have to, so I'll give you like letters and you'll have to pick a letter to tell me what this thing is or whatever. Okay, there'll be some of those things. So really when you think about it, there's gonna be matching, but the matching will be kind of like images and you'll have to tell me what those things are, okay? And so this is a review that is a general review. It's not specific about anything. However, I will be very happy to answer specific questions that you might have about anything that is within the realm of this particular exam. So the exam is just those 3 topics. There's a lot of stuff in there, okay? So here's an example of a multiple choice question. When 5% sheep red blood cells are added to a medium, allowing clinicians to discern whether the bacteria can destroy the blood, the medium is considered A, selective, B, enriched, C, non-living, D, differential, E, A and B, A, B, C and B, or A, C, D and B.
E.
So D and B, so it's A, C. So if we were doing this by Scantron, I have got enough questions now that I can put it on computer. So I think that's the way I'm gonna do it, so I don't even have to run a Scantron. I'm gonna run it through Blackboard. I've got one from last semester that I'm probably gonna use, but I'm gonna take out some stuff and put some stuff in. I may or may not, in that process of taking and putting, I may or may not have duplicated some questions. I don't have time to go back and check them all. So if you get the same question and you know the answer, you're just gonna be hugely benefited by it. However, if you get the same question and you're not sure what the answer is, then maybe you answer one way on one of them and one way the other and have 50-50 your chance of getting it correct, okay? So because it's gonna be by computer, you can use the computers in front of you. I'm not gonna use monitor, I'll be in the room, so if you've got anything that you need clarified, I can come over and clarify it for you. But there's over 400 questions, you're gonna get 60 of them. So it's gonna randomize everything, so therefore I don't really care where you sit because the questions are gonna be different for everybody, right? I would prefer that you didn't sit in that back row because I don't like it when I can't see what's going on. So take up all of the space out here first. You can bring your own computer, I don't have a problem with that, okay?
So the answer is enriched and differential?
That's correct. Okay? Any questions about the setup? If I feel like I can't use the computer for whatever reason, then I'll just make it paper and everybody will get the same questions. But I think right now I can make it so that it can be facilitated through Blackboard. I'm not gonna use monitor, I'm not gonna watch you, huh? Okay, here we go. Sterilization of bacteria, that's the prime. So be sure when you're taking a multiple choice, you read the question very carefully. Don't immediately jump to conclusion, right? So sterilization of bacteria may be accomplished by the following except, which one of these is not a sterilization process against bacteria? A is correct. Pasteurization is a sanitizing method, it's not a sterilizing method. Every one of those other things is a sterilizing. I'm being very specific there with a 0.22 micron filter. Remember, a 0.22 micron filter, I said, will sterilize for bacteria, but not for viruses or prions. But this is specific for bacteria. A microorganism that grows optimally in our body is termed?
Mesophiles.
That is correct, it's A, mesophile. Oh, by the way, these questions I just took from my question pool. They're real questions, right? You may get one of these, you might get all 15, right? You'll just be lucky if that's the case, but there's gonna be others, okay? Detergents with a 10-minute contact time kill microorganisms by? Huh?
The plasma B.
B, by solubilizing plasma membrane, that is correct. Which of the following describes flash pasteurization? Which one? B is correct. Mhm. See, y'all are having no problems with these, so why do you think this is gonna be hard? Yeah. Anyway, AUG codon functions in coding for the amino acid methionine and as a, which one? B, start codon. Mhm. What's wobble? The last nucleotide in a codon can be different, but it still codes for the same amino acid. There will not be any multiple choice on your part because it's all multiple. I mean, there won't be no fill in a blank. But let's just go through this because I might have this and then just make it multiple choice by putting the answer there, right? The answer will be right in front of you. There'll only be one answer, so all you have to do is pick the right answer. So if you know the exact ingredients of a medium, it's termed what? Chemically, not chemical, chemically defined. Okay? In general, a something is a sanitizer that is too toxic and should not be used on the skin or mucous membranes. That's specific, in general. Something that's dangerous, starts with a D, ends with a T, disinfectant. Something is a term used to describe two microorganisms growing in an environment where one of the microorganisms benefits from the relationship, but the other microorganism is neither harmed nor benefited. Huh? Symbiotic is general, specifically which one? Commensalism. So, because they're multiple choice, like for instance, I might ask something like, which one of these, like I might have a picture of something like Lysol, and then I'd say, which of them does it fall into what category, right? So A, phenolic, B, alcohol, C, halogen, D, detergent, or E, alkylating agent. So I can do 10 questions on just that kind of stuff, right? There'll only be one answer. Okay, so be sure you know the representative that's on the PowerPoint. Those are the ones I'll use. I took those exact pictures and stuck them in as the thing on the exam. I've been working on this for a long time, y'all. So I think it's gonna work just really great. We'll see. Okay, so you're gonna have something like this, except you're gonna have to do the math and then you're gonna have to answer the question because it's multiple choice, right? So for instance, if an egg salad sandwich sitting in a warm car for 3 hours develops 10,000 salmonella cells, how many more cells would with just 1 hour more in the car be generated? So how would you figure this out? The final population is equal to 10,000 times 2 for binary fission. How many generations?
9.
No. The 3 hours is gone already. It's 1 hour after that. So if the generation period for salmonella is 20 minutes, it's 1 hour, 3.
3.
So that's the answer. But of course, you're gonna have to get a final answer. So that's 2 times 2 times 2, which is 8, is 80,000. And that's the one you would click.
Just 1 more hour.
Be sure you read the question, okay? Because people will add 4 and 3 and 1 and get 4, and you'll go crazy with numbers, and you're like, probably I've figured this out, but that number's not even here, right? Or the really bad thing is I include that number as a choice, and then you're like, oh, I got it right, and then you didn't read the question. Be sure you read the question. Describe how an amphipathic detergent could be used as a chemical control agent, but instead is used only as a mechanical control agent.
Polar and non-polar, and one's attracted to therapy and attracted to one.
Yes, exactly like that. But on a multiple choice, it would be something like the molecule has a polar and a non-polar end, something like that. Has to be real simple. I can't make the answer this big because, you know, usually
The long ones.
the long ones are the right ones, right? So I have to make them kind of, and pay attention. I try to make them all kind of the same amount of words. So be sure you understand the words, okay? I can't go crazy on a multiple choice, right? It's a quick little, you're gonna get 70 minutes to answer 60 questions. That's plenty of time. And when you take a multiple choice exam, you never leave anything blank. If you leave something blank, it's automatically wrong. If you don't know, guess. And if you don't know, use deductive and inductive reasoning to figure out. It's not A, it's not D, I don't think it's C, so it's either B or E, and now you're 50-50. And if you have to go flip coin, okay, that's what it is.
If we do on the computer, we can go back, right?
Yeah, so I'll arrange it so that you can see all the questions all at once. And there'll be a timer, and then there'll be like, I don't know if you've ever done Respondus, but there's a little block, and every time you answer the question, it shades in the block, so the only ones that are not shaded in are the ones that you're missing. So therefore, you don't have to like figure out which one I'm missing, right? It'll tell you which ones you're missing. And the way you take a multiple choice exam, if you've never had a strategy for it, is you read the question, and if you know it, you answer it. If you don't know it, you don't sit there and think about it, you let it go and you go to the next one. Then you keep going until you get to number 60, and then you go back. And because that way you have however much time it is left over to think about the ones you're not sure about. And once you make a decision, don't change that answer unless you read something else, like, oh, I was wrong on the other one, then go back and fix it. But usually you don't wanna change your immediate response, because usually that's the one that's correct.
So, is it 70% of this multiple choice? So how are we gonna calculate 60 questions with the 70?
It's just 60 divided into 70.
1 point something like that.
Slightly 1.1, maybe not even, maybe 1.08 or something like
You're
that.
not gonna make this
meeting after we figure it out, or will it?
For the multiple choice part, yes, but I have to look at the other stuff, right?
Yeah.
So you'll know that part very quickly, and you can go back and review if you want. I'll give you a 15-minute window that you can go back and you can't take pictures of it though, right? But you can go back and review it. You just can't, once you review it one time, you're locked out forever.
Can we
Unless you
get
come to me and say, I wanna review my exam, then you and I can sit down, I'll open it up for you. Yeah,
Do we get scratch paper?
yeah,
Okay.
I'll bring it to you, okay? Plus a codon table, yeah. You can bring your calculator if you want, if you feel comfortable with it, all right? And if not, I'll have some simple calculators. Good. You can't really program anything in there. Well, I guess you could program the population formula, and then you can put, if you're really smart. Well then, to me, if you're that smart, you deserve the credit for being able to formulate that formula into your calculator, and I'm good. But you can't use a computer. You can't deviate from the computer that you're using. If you feel more comfortable with your own computer, I have no problem if you want to bring your own. If you want to bring your own tablet. No. Fine. Okay. Okay. But if you're more comfortable with your tablet, Daniel, you can use your own. I have no problem with that. The only thing that doesn't work all that well are Chromebooks. So if you have a Chromebook, you might want to use my computers. Okay, because sometimes the images. With a Mac also, sometimes my images will not show up. There's a little window, you have to click on the window to see it, but that takes too much time. So if you've got a Mac, I would almost prefer that you use the computers here because you'll have more time. Okay? Good? All right, so here, I might do something like this, but of course it'll be multiple choice, so I have to figure out how to do that. But why would chlorine be considered more effective antimicrobial agent than ethyl alcohol? So you have to think about this. Remember, you have to normalize it because they're not in the same manner. This makes a really good question if you're doing short answer. It doesn't make a really good question if it's multiple choice because I have to normalize it somewhere or another, right? But if you think about that, if you normalize it, what would be the answer? It's about contact time, right? So if we look at this, you could see the answer that I would be expecting are, if we look at the actual data presented by chlorine, and if we normalize it to maybe make it 10%, we would need less time of contact for the organism to be killed by chlorine when compared to ethyl alcohol because it takes minutes for there. Okay? And then use the above table to explain contact time using alcohol necessary to control gram-positive and gram-negative bacteria. So now you're only looking at this data, and it says that gram-positives need 10 minutes, where gram-negatives need only 2 minutes. And the reason that is because of the LPS layer. The alcohols are much more attracted to the lipids than they are the bacterial glycan. Okay? So, there can be matching or there can be multiple choice. I can turn these into all kinds of things, right? So I'm pretty creative when it comes to test writing, and you'll see that creativity when you look at the test. But there's gonna be some things that are gonna match up with these, and I can't fix it because this was a picture I took, and when I took the picture, I didn't recognize that it was off kilter, so it's off kilter, so I have to go back and fix that at some point. But here, what matches with diamine? Over here, what matches it? Contains 2 nitrogenous rings? No, that's not it. What is it?
Is it
D. A DNA nucleotide, that's correct. Who said that? You, okay. You said it too? I'm sorry. Okay, ligase, what does it do? A and B. AD, that's correct. What about bacterial DNA? AC, 1 chromosome. Semi-conservative replication. D, DNA nucleotide, no. E, my bad. And I have my hearing aids in. Parent DNA strand used as a template, correct. How about UAA, UAG, UGA? AE, protein synthesis complete. This is termination sequence, right? What about 5' to 3'?
AB.
A, which one?
AB.
AB, DNA replication direction. Purines,
DNA replication direction.
yes.
Oh, the off thing is messing
Purines,
up.
B, contains 2 nitrogenous rings. Helicase,
It's the unwinder.
unwinds DNA, A. Thymine dimer. This takes 2 concepts into consideration, right? Control and DNA. Is BC, ultraviolet light DNA mutation. Right, and polymerase. Polymerase, copies. So C, copies DNA. So then you're gonna have to do this, right? And when you do that, we've done it already. This is the one that was on the board. I'm not gonna do it again. But I'm probably, because it's multiple choice, I'm not gonna give you a big old gigantic fragment. I can do this lots of different ways, right? So I can give you a codon and ask you to tell me what amino acid it codes for. I can give you a small fragment, maybe 3 nucleotides that are DNA, and ask you for the codon, right? I can ask you for the anticodon. There's lots of ways I can do this. Okay. One of the easiest, and then, so, let's be turned into multiple choice. Also describe the DNA replication as it relates to the following: leading strand, leading strand, 5' to 3' continuously, lagging strand, backwards in fragments, Okazaki fragments, and semi-conservative replication, 1 strand from the parent DNA conserved for each new DNA molecule.
So that
Just
one, instead
like-
of rewriting, give an answer, we can
It's
just
gonna
pick.
have to be multiple choice, right? And so I'll have to figure out what that looks like. Okay? And then here's another way I can do this. This is blood agar. Is it selective differential or enriched or a combination of there?
In combination.
here is a standard growth curve, but I want you to know it this way. And I've already developed this, I have the standard growth curve, and I've got some letters on it, and then I ask you different things, right? Largest population, where would you find that? C. I might say endospores germinate, and you'd say what? A. I might say things like logarithmic growth, and you'd say
Rob?
logarithmic growth, B, okay? So it's gonna be something like that, okay? And then I'm gonna use this also, right? So what represents 1? Topo isomerase, so very good. What represents 5? 5 is the light pink, not the dark pink. What's the light pink? It's placing something into the DNA molecule. What do we call that enzyme? Am I in your way? What do we call that? Primase, right? If we ask for the little thing that's labeled 4, the dark pink thing, that's the primer, okay? 8, what's represented by 8? That's that fragment right there. What do we call that? Okazaki fragment. And then my A's and come out. This would be A and this would be B, or A and B. If I said A here, what strand is that? The leading. And B, what strand is that? The lagging. And then looky here. So what represented by A? What's represented by
That's
A?
the backbone.
The backbone. What's represented by B? Now you have to look at how many hydrogen bonds are here. How many?
But 3.
3. Therefore, that's the big one. And what is it? Guanine. This is the small one. What is it?
Cytosine.
Cytosine. Very good. What's this thing?
That's a
Phosphate. What if I said, how would I do that? I can't do that. Fine. Okay. What if I said E? What is that pointing to?
It's specifically oxygen.
Yeah,
Your pointer was
it's
pointing
pointing
there.
to that little.
Oh, are you talking about the ring?
It's a sugar, right? So if I was to do that, I would say sugar or hydroxyl. That looks kind of in the middle, so I would only have one of those as a example, or as a response. I wouldn't have both. That'd be not fair. Okay. What's represented by F? So you need to understand how many hydrogen bonds are there.
Amine.
2. So this is thymine, and that is adenine. Very good. So this makes perfectly good sense. I can make all kinds of questions from this one image. Plus, I can make a whole bunch of questions off of this image too, right? So this is the process. If I have a big bracket, it's a process. So the process of copying DNA into RNA language is called what? Transcription. If I'm looking at this particular molecule, what is it? Messenger RNA. If I'm looking at this process here, what is that called? Translation. What about this little firecracker looking dude? That's a tRNA. And what about these little things that have different shapes that are being put into a series, or you could call it a peptide, but if I didn't have, what if I had an amino acid as the response? You'd have to pick amino acid. So you want to be sure that you can understand it can be either one of those. Okay? I'm only going to put one that's right. Every question will only have one correct answer. And if you're confused, you can call me over. I can help to try to clarify. Okay?
So you're saying that you would more be looking for it saying it's an amino acid rather than
I don't know. I
peptide
mean,
chain?
I can do so many things with these things, right? So there's just all kinds of different ways I can ask them.
Would you do something like you'd have a peptide chain and polypeptide chain?
No, I would never do that
Okay.
because I don't even know if I told you what the difference was. I did,
Yeah.
but I still would do that. That's just downright mean. Yeah, I'm not here to be mean. Okay? Let's go to lab. It's time for lab.
Do you guys remember the picture with the